Welcome everyone to part 3 of day 1. This is a session is all about experimental techniques regarding single cell, and we have 3 great speakers. Our first speaker is Luis Saraiva. He is a principal investigator from Sidra Medicine, Doha, Qatar, and he will talk about using spatial transcriptomics to unwrap the anatomical logic of smell. Luis.
Okay, thank you so much. I guess you can hear me and see me. I'm going to start with a slide of the small town where I'm from, originally in Portugal. This is where I grew up and where I explored a lot of the different areas of these mountains all around. As we explore nature, we notice that we encounter many different sounds, many different smells. These smells can come in various different shapes and odors. It's known that animals can perceive many of these chemicals as having a very distinct odor. It's also estimated that humans can discriminate anywhere from 10,000 to 1 trillion different odors, depending on which estimate you believe the most. Of course, some of these odorants, we know that they are a major component of foods and flavor, while others may signal the presence of danger, other animals, etc. Now, we know that evolution ensured that different animals end up with very different noses and olfactory receptor repertoires, which help the different species to perceive and respond to external chemical, to their external chemical world in a very unique way. This is, of course, critical for their survival and reproduction. In the whole olfactory mucosa, the whole olfactory mucosa sitting within the nasal cavity is the organ that allows us to smell. Certain species, certain mammalian species have an additional organ, which is called the vomer nasal or Jacobson organ, which we'll not be focusing on in this presentation. Like most tissues, the whole olfactory mucosa, or WOM in short, is a complex tissue composed of many different cell types organized in a stratified manner. You see that the basal layer is populated mainly by globous and horizontal basal cells, which are stem-like cells, the olfactory and sheathing cells, which are glial-like cells, whereas the middle layer is composed by immature and mature neurons. The mature neurons extend their cilia out in the mucous layer, which is in direct contact with the external world. And of course, the medial and the apical layer is also populated by sustentacular cells, which are the cells that keep the whole epithelium in shape. Our understanding of how the olfactory system works was revolutionized with the discovery of the olfactory receptors in 1991 by Linda Buck and Richard Axel, who won the Nobel Prize in 2004 because of their discoveries. Of course, for us, the most interesting cell type of them all in the olfactory mucosa are the olfactory sensory neurons, or OSNs in short, which are the functional units of the olfactory system. And why? Because they express olfactory receptors in their cilia. These olfactory receptor family is highly conserved between invertebrates, with about 135 members in zebrafish, about 1,100 intact olfactory receptors in mouse, and about 400 in humans. Now, how are these receptors expressed in the olfactory mucosa? Well, work done by the by the Buck and Axel labs later on found that each olfactory sensory neuron that populates the olfactory mucosa of the nose chooses to express a single allele of one intact olfactory receptor gene. They also found that that different areas of the olfactory mucosa are populated differently by different olfactory receptors. This means that olfactory sensory neurons expressing the same olfactory receptor are randomly dispersed within a single zone of the olfactory epithelium. You can see, for example, that this yellow olfactory receptor is expressed only here, as you see by the yellow dots. The red one is expressed by this band here, and so forth. If you can summarize these zones across the olfactory epithelium of the mouse, this is a coronal section, a schematic of a coronal section. You see that they are expressed in different concentric rings, starting from the medial position. But how heterogeneous is the olfactory system at the molecular level? Well, some of my work that started in 2013 aimed at answering these questions. And for this, I actually first started comparing the mouse and the zebrafish olfactory mucosa. This comparison spanned 450 million years of evolution, and later expanded this work to a wide range of other mammalian species, such as human, macaques, marmosets, rats, and dogs. Within the mouse, we also performed RNA sequencing in pools of isolated olfactory sensory neurons and also in single cells. Now, in the interest of time, I will not expand much on this topic, but I will like to leave you with 6 key results that emerged from this work. Number one, RNA sequencing, as you know, is a powerful technique that can aid in the discovery of rare, novel, and cell types. And in this particular case, by doing single-cell RNA sequencing, we found a novel cell type of neurons that's conserved between mouse and zebrafish that expresses Gucci1b2, which is a cytoplasmic protein, and that later work found that this population of neurons is actually responsible for detecting the concentration of O2 in the air, which allows the animals to avoid areas with low concentrations of oxygen. Number two, olfactory receptors are expressed across a large dynamic range, with a few expressed at very high levels, and with many expressed at low levels. Here you can see the log10 expression of the olfactory receptors to visualize the whole distribution a bit better. Also, olfactory receptor distribution across the whole olfactory mucosa is due to the number of OSNs expressing a given olfactory receptor, and not due to the number of olfactory receptor transcripts in each OSN. This is promising because it will allow us to use olfactory receptor expression as a proxy of OSN quantification. We also found that this type of distribution is conserved across all of the mammalian species we've analyzed. And we also found that highly abundant olfactory receptors vary greatly among species. So you can see these ones here that are very highly abundant in mouse and rat, whereas these ones here are, for example, only highly abundant in humans, etc., etc. Also, we found evidence for a detection bias in highly abundant human and mouse olfactory receptors towards ecologically relevant odorants, such as food odors in humans and pheromones in mouse. Next, my work took me towards the application of applying spatial transcriptomics to deconstruct the anatomical logic of the olfactory system. For this, we started applying a transcriptomics approach to study the spatial molecular heterogeneity within the mouse nose. For this, we established a collaboration with some computational scientists at the Helmholtz in Munich, and with a group at the University of São Paulo, which helped us with the validation. The experimental approach was fairly straightforward. First, we started cryosectioning the whole olfactory mucosa across the 3 Cartesian axes: dorsal ventral, lateral medial lateral, and anterior to posterior. Next, we performed RNA extraction from each of these cryosections and performed RNA-seq in each one of them. The first thing we looked at was to look at genes that we know are expressed in a zonal fashion. For example, the ACSM4, we know that it's a dorsal known marker, and as you go from the most dorsal to the most ventral, you see a decrease. Whereas REG3G, we know that it's a ventral marker, and as you go from the dorsal to the ventral position, you see that its expression increases, validating our method. Of course, you can then look at the expression levels of these genes across the other axes that we profiled as well. The next thing we did was quite obvious. We started calculating the genes that are differentially expressed in space, and this resulted on the identification of many different zonal patterns of expression for both olfactory receptors and other genes. You can see here the differential expression analysis within the dorsal ventral axis, and you can see the teach dot here corresponds to a particular expression pattern here from the most dorsal to the most ventral axis. And you can see that we identified 9 broad areas of expression within
4 minutes remaining.
9 areas in the AP axis and 5 areas along the lateral medial axis. We also then use this data to reconstruct the gene expression patterns in 3D. And as the ACSM4 reconstruction shows, we see that this gene is mostly expressed in the dorsal axis. We also then did this for every single gene that is annotated in the genome, and we performed some in situ hybridization to validate the predicted patterns. We then really focused on the expression of the olfactory receptors, and I'm not going to go into details because this work is also published, and in the interest of time, I don't have time to elaborate a lot, but we used LDA, an LDA algorithm on the 689 spatially differentially expressed olfactory receptors that we detected among all of the different axes to define broad areas of expression. And we identified 5 broad areas of expression consistent with some of the previous work that has been published. We then use this in combination with machine learning to predict the gene expression patterns in space for all the other olfactory receptors that were expressed at very low levels or that were not detected in the RNA sequencing expression. And we, of course, performed a bunch of validations that really allowed us to provide accurate 3D spatial indices for the entire repertoire of olfactory receptors, which represent for the entire repertoire of all of the olfactory receptors annotated in the mouse genome. Last but not least, we decided to tackle an important question that has been unknown so far. We know that for most sensory systems, the functional logic underlying the organization of primary receptor neurons and their receptive field is known, but the anatomical logic of smell was still unknown. So we decided to go into the literature and retrieve all of the odorant ligand pairs that have been described for mouse olfactory receptors, and we ended up with 738 olfactory receptor ligand pairs. Among these, we found that the ones that among the olfactory receptors that share ligands, they have a similar spatial index, and this really is consistent with the hypothesis that olfactory receptors, their zonal distribution depends at least partially on the properties of the odorants they bind to. Then we basically retrieved around 1,200 physicochemical descriptors for each of the ligands, including the molecular weight, the number of atoms, et cetera, and computed a Spearman correlation of each of these descriptors with the average 3D index for the receptor that detects them. And we did find a statistically significant correlation for 744 descriptors. And amongst the 5 top descriptors, we realized that all of these are related to odorant solubility. We found that we also found that when we map this, the average olfactory receptor 3D index to the top descriptor, which is the air mucus partition coefficient, which is a physicochemical descriptor that predicts how much a particular odorant is diluted in the mucus, we found a positive association between that and the olfactory receptor, the spatial olfactory index of the olfactory receptor that binds it. And when we map some of these receptors to the nose, use their expression in the nose, you see that, for example, receptors that bind hexanethiol are expressed in the most ventral lateral area, whereas receptors that bind, for example, 8-bromo-octanoic acid, which is highly soluble, are expressed in the most dorsal part of the epithelium. These results show a direct association between the olfactory receptor spatial patterns and their calculated absorption patterns, providing a potential explanation as to why behind the physiological function of these different zones that we found in the olfactory mucosa. Last but not least, we provide this atlas online. It's free to browse if you're interested in olfaction. The link is in the paper, so you can just go and look it up. Thank you very much, and I'll take any questions if there is time for that.
All right, thank you, Luis. So we'll wait for the questions since we have 1 more minute. So I'll just start asking a question. Coming from the genetic background, I'm just wondering, olfactory receptor have very many mutation in different ethnicities, how that contributes into the cell types and the heterogeneity of it.
Yeah, that's a great question. So when we do these experiments in animal models, we can control the the genetic background, right? So what we see is that when we perform these experiments in isogenic strains, we see that the expression of the different receptors is fairly maintained between different individuals. When we looked at humans, so we profiled humans, from different the average variation in humans is higher, and we see a little bit more variation. But if you take the top 10 percentile of most expressed genes in an individual, that percentile of genes will also be the top 10% in the other, even if the order might change a little bit. So that's relatively conserved.
Okay, so you have two more questions, but I will, ask the questioner to directly reach out to you. For the sake of time, we'll go to the next speaker. Thank you, Luis. So our next speaker is Binte Ashraf. He's a postdoctoral fellow at Mohammed bin Rashid University of Medicine and Health Sciences. And Zahra will talk about integrative analysis of long isoform sequencing and functional data identifies distinct neuronal subtypes derived from human-induced pluripotent stem cells. Zahra, please.
Hello, everyone. Is my screen visible?
Yes, yes.
Yes. Hello, everyone. Good evening. This is Binte Zahra, and I'm working as a postdoctoral researcher at MBRU. And I would like to thank HCA to give me an opportunity to present my study here. So today I'll be discussing that how single cell has privileged us in identifying the neuronal subtypes denoted by iPSCs using differentiation protocol that we have established in our lab. So if we go back and go back to 2006, so the advent of iPSCs was a groundbreaking discovery at that time, and it has totally revolutionized the field of research. So when it comes to study model, it is very critical to use an appropriate study model that can exactly recapitulate the test condition with that we want to study. So in this regard, iPSCs has been proven as the most convenient model because they can be differentiated to various cell types by introducing transcription molecules to the culture conditions, leading to the activation of stimulus that can lead to differentiation into the specific lineage that you are interested in. So the same way with the use of specific neurogenic stimulus, we can activate ectodermal lineage in iPSCs, thereby producing neuronal progenitor cells that can be further differentiated to neurons and can serve as a study model for any neuronal disease. However, there are several protocols that has already been reported in the literature, but we still face the major challenges of low efficiency, presence of undifferentiated iPSCs in the culture system after differentiation, and of course, the time required to generate fully functional neurons. However, there are several methods that can be used to manage the drawback of presence of undifferentiated cells in the culture. We can use suicide genes or suppressor genes. We can even go for FAG cell sorting to particularly using marker antibodies and particularly picking up the cells which are the neuronal lineage. But all these are expensive and requires expertise. There is another conventional yet economical method that can be used as well, that we can manually check for the undifferentiated cells within on the petri dish using the microscope, and those can be eliminated by manual scrapping. However, this technique is convenient, but it requires technical expertise. So, keeping these in our mind, we have modified the existing differentiation protocols to improve the efficiency, time, and also deal with the growth of undifferentiated cell iPSCs in the culture. So here we are showing that how iPSC culture conditions impact the efficiency of neuronal differentiation over the time. What we actually did, we used two different culture conditions to grow and maintain iPSC. One was basal media system and another was differential. Taking iPSCs from these two culture systems, we first induced ectodermal lineage, and then these NPCs were differentiated to neurons. However, NPCs that were generated from basal culture system, that is stemflex media system, needed to be expanded till Passat 7 before we can go for a neuronal differentiation. However, the NPCs which were generated from the other system were morphologically distinct and were ready for the differentiation to the neurons. Also, it affected the timeline from a stemflex media system. It took almost 60 days because of that expansion till their P7, Passat 7. And for the other system, it was not needed. So within 25 days, we were having functional neuron in the culture dish. So when iPSCs were shifted from respective culture systems, culture media to neuronal induction condition to start to generate NPCs, we observed different morphological changes in the cells. So here we are actually showing that how morphology of iPSCs is being changed over the time and to generate NPCs. So we observed that day 1, day 3, and day 5 was kind of similar for both culture systems. However, on day 7, in stemflex media system, there were many undifferentiated cells growing with NPCs, while those as indicated by the red arrows in figure G. However, those cells were not there in the other culture media system. We observed only NPCs growing in the culture dish as indicated by black arrows in figure H. So here now, once we had NPCs, we started differentiating them to neurons. So we used different, in the first panel, which is panel 1, we used different passages, and we used different passages to show that how the morphology of NPCs were varying between the two systems. And it can be seen that from in E8 media system, the cells were having neuritic extensions till even Passat 7, and looks ready for the differentiation to neurons. However, for the stemflex media system, we couldn't see those neuritic extensions until Passat 5, but also during the course of these passages, we observed the growth of undifferentiated cells in the culture dishes. Then we used Passat 2 cells to start neuronal differentiation from both culture systems, E8 and stemflex. And we found that by day 15, the cells were fully differentiated to neurons, as it can be seen morphologically that they were having neuritic extension. But in the case of stemflex media culture system, those neuritic extensions were not there. And even till day 15, we couldn't see anything. And when we passaged them after day 15, we found that the cells look like more of a neuronal progenitor cells, while for E8, there were proper neurons growing. So here we took Passat 7 cells, because for a stemflex media system, we need to expand them.
3 minutes left.
Okay, so we wanted to passage. We wanted to see how cells are differentiated to neurons in stemflex media system. So we used Passat 7 cells and differentiated them to neurons. And we saw that in the stemmatic media system, the cells were completely differentiated. However, there were lots of undifferentiated cells in the culture dish. So then we did phenotypic characterization with the cells derived from E8 media system, and we found that, and here the distribution plot shows different somas and neurites and terminals. Then we did molecular characterization using immunofluorescence with marker antibodies, and we found that NPCs were positive for all the NPC markers. Those were SOX2 and nestin, while the neurons were negative for a NPC marker, but positive for neuronal marker. Those were MAPT, NUN, and synapsin. We also did functional characterization of these iPSC-derived neurons from E8 system, and we measured electric potential measuring patch clamp system. And we found that the cells were generating action potential over the time, and also by changing the different electrical current, we observed generation of current in the cells. So this confirmed that the cells were functional, the neurons were functionally active and were having electrical activity after day 25 of the differentiation. Now comes the most interesting part, that is the that we used single cell RNA-seq to see what kind of neuronal populations are present in our in this neuronal cell population that we have established using this protocol. So we found that there were multiple neuronal populations that were generated from those NPCs that were coming from E8 culture media system. Taking the advantage of long read sequencing, we also looked for the looked into the diversity, that how diverse isoforms are being expressed in the cell, how diverse isoforms of cell marker genes are being expressed in cell population, and we found that there were multiple cell isoforms that were expressed in the cells, with PEG6 gene having more than 10 isoforms expressed in the respective cluster.
2 minutes.
Lastly, we identified cell pathways that were upregulated in iPSC-derived neurons in comparison to the NPCs, and we found that those are the pathways which are were upregulated belong to neurogenesis, synapses, and neurodevelopmental pathways. So lastly, I'll conclude that I'll conclude my talk saying that by this study and utilizing single cell, we were able to see how different cell populations are being expressed from the homogeneous population of NPCs, and also we were able to identify the signaling pathways that are associated with the regulation of neurogenesis and neuronal differentiation from NPCs to neurons. Lastly, I would like to thank my university, MBRU, for providing us this facility to conduct the research smoothly. And I would also like to thank my PIs for their incredible support and encouragement over the time, and the team who supported me to establish this protocol and to do the analysis, especially Nasreen and Kumail for helping me in the analysis. So this is our research team at MBRU, and these two are my PIs, Dr. Mohammad Addin and Dr. Bakram.
Thank you, Zehra. I'm just wondering if there is any question for Zehra. Let's see.
Yeah. Maybe I can start.
Sure. Yes,
yeah.
Naomi.
Hi, thank you for the lovely talk. It's very exciting. I was wondering if you considered comparing your signatures of the differentiated neurons to some of the published cell atlas in the developing neurons to see how much. I mean, it was really impressive to see their functions. So they're definitely neurons that are active. Wonder how different or similar they are to neurons in developing brain.
No, we didn't conduct any study till like that how, but we did only the single cell to see what all cell types are there, and yeah, what the two distinguish those cell types or to identify those cell types in the population, we use the marker genes which are published and which are present in these databases.
So you basically use HCA databases as well.
Yeah.
For the marker. Okay. All right. So if there is no further question, I would like to thank Zehra for your lovely talk. And I'll go invite the next speaker, who is Richa Naveed. Is a postdoctoral fellow and a data scientist at the Center of Applied and Translational Genomics in Dubai. And Richa will talk about delineating the CHD, the congenital heart disease linked cellular heterogeneity using mutation enrichment analysis. Richa.
You see my slides?
Yes.
Okay. So, hello everyone, I'm Richa. Thank you for the introduction, Dr. Mohammed. I take this opportunity first to thank Human Cell Atlas for giving me this opportunity to present my work at this forum. So I'll be talking about, delineating the CHD linked cellular heterogeneity using mutation enrichment analysis. So congenital heart disease is one of the most prevalent neonatal congenital anomalies. It affects the structure and functioning of the heart, and it is one of the main reasons for morbidity and mortality in children worldwide. So it is not just one disease, it is a big umbrella which consists of many different types, many different phenotypes, such as tetralogy of Fallot, in which there is abnormal heart and sinuses, then there is cohesion of aorta, in which there is narrowing of aorta, thereby reducing the blood supply, affecting the blood supply to the rest of the body, and so on. So, and then there are many different factors, such as environmental factors and genetic factors, which can lead to CHD. However, in a majority of cases, it is the reason for CHD is unknown. So this was the main aim of our work. So we wanted to deep dive into the genomics of congenital heart disease. And we wanted to look at the clinically relevant mutation, identify and prioritize the CHD genes, and then to identify if there's a link between genotype and phenotype, and if there are some specific cell types associated with those genetic mutations. So this was the overall aim. And I'll very briefly tell you what we did in this meta-analysis. We started with 16,349 variants from 3,166 samples from the literature. We did variant annotation, and then we did ACMG-based classification, identified the pathogenic and VOUS mutations. We then took them for downstream analysis. Then we identified the priority genes, and we identified 90 priority genes, and then we used those genes for characterizing the phenotype, for pathway analysis, and for cell type mapping. So I'll be discussing these steps as I show you the results also. So now here, these are the basic descriptive analysis of the 16,000 plus variants. So they belong to so they were from 3,100 samples, which had different phenotype. They were distributed across all the chromosomes. Majority of them, 83% of them, were loss of function mutations, and 17% of them, 70% of the mutations were de novo. 4% of the mutations were recurrent. They occurred multiple number of times in unrelated individuals, and their distribution is shown over here. So then after this, we did the pathogenicity analysis using ACMG criteria, and we discarded the 0.1% of benign mutation and moved ahead with the pathogenic, likely pathogenic, and VOUS mutation over here for the next downstream analysis. So this set of mutation which we used for downstream analysis were around 15,000 variants, and they were distributed across 8,000 genes. So of these 8,000 genes, we used in a stepwise manner. We identified the genes which had high probability of causing CHD by narrowing out the genes which are present in multiple unrelated patients. The mutations can be recurrent or non-recurrent. So that was 90. So these 90 genes were present in 3 or more than 3 patients. So then we constructed our own in-house CHD database combining the different CHD panels and available CHD genes, gene list in the literature. And we also took the OMIM genes and we overlapped these 90 genes with the OMIM genes. And we identified that these 6 genes are not having very strong relationship with any of the diseases. So these are novel genes. So then we took the top 10 genes, depending on the number of samples, number of patients in which they were mutated, 15 or more. So we took the top 10 genes and characterized their mutations in terms of what kind of mutations are there, familial, de novo, and LOF, and so on. And after that, we moved to single cell data. So obtaining pediatric samples and carrying, it is very difficult, and that is the reason we have only a single, till date, only a single CHD single cell experiment available, and luckily it was published when we needed it. It is a beautiful work by Hill et al. In Nature, and this so there are 6 CHD samples within the age range of 2 months to 4 years, and the control samples between 4 years to 11 years, and we use this expression matrix from this DO. So it consisted of 53,208 cells in the CHD and 54,261 cells in the control data. So we reanalyzed this data, we remapped the UMAP. And so just before going to the results, I'll just show you the CHD heart and control heart UMAPs. So they're very similar. However, the majority of it, more than 50% of cardiomyocyte cell types were present. And you can see there is the difference between the cardiomyocytes from the heart and from the CHD and control heart. So then we first analyzed the expression of top 10 genes. So these top 10 genes were more expressed in CHD cardiomyocytes. And we also did the subtype analysis. We subclustered the cardiomyocytes. And so here you can see there's a difference between the clustering. So we developed our own algorithm in our lab for the subclustering, and you can see that there are 5 more clusters present in the CHD heart, which is not present over here. And there was a ubiquitous, more or less ubiquitous expression of the top 10, the combined expression of top 10 genes across these. So next, we moved on to the top 2 genes, which is Notch1 and MYH6. Notch1 was expressed in 60 patients. MYH6 was present in 36 mutations were occurring in 36 unrelated patients. So we took this and first we tried to see if there's a link with phenotypes. So 67% of Notch1 mutations were present in TOF cases, and MYH6 more than 80% were present in quotient of aorta cases. Then we also found that Notch1 was more expressed in the endothelial and endocardial cells, and MYH6 was more expressed in the cardiomyocyte cells. So how we identified this is on the basis we took into account both the fold change in percentage of expression as shown over here, and which shows higher expression specifically in the endocardial cell. So next we compared the expression of Notch1 in endocardial cell between the CHD and control pseudobulk analysis from the single cell data. So we identified that there's higher expression in control, significantly higher expression, and similarly MYH6 was also more expressed in control. So possibly, these patients,
5 minutes.
These patients do not have a mutation in Notch1 or MYH6 because that data was provided by Hill et al. So possibly there's a gene-to-gene, some genetic interaction going on, which downregulates the expression of these genes in CHD cases. And then we also did the subset analysis for MYH6, and we identified that MYH6 was downregulated in these 4 clusters and in the CHD, and it was downregulated in these 2 clusters, these 2 cardiomyocyte subclusters in the control data. So then we also we took all the 90 genes and we did the network analysis, and as expected, we observed a very high enrichment in the heart development pathway network. So for this, we had used gene overlap package. I did the enrichment analysis in R and then used cytoscape to produce this figure. And the database is a pathway database which we use WCAG and gene ontology databases. So apart from the heart-related pathways, signaling pathways, transcription pathways were also involved. So this was as expected because most of these genes are involved in formation of heart at an early stage. So finally, I would like to conclude my work. So this is the first comprehensive study linking CHD genotype, phenotype, and cell type. So in this study, I identified 90 mutation-enriched genes, of which 6 are novel candidate genes. So these 6 genes are not, they were not very well expressed in the single cell data. The reason which I think is most of these genes are, there's very less functional annotation for these 6 genes available. That's one reason, one thing. And second thing is because they are not cardiac related genes, they're non-cardiac genes. So we are planning to expand in that direction to check the role of non-cardiac genes in congenital heart disease. And then we also identified here that the frequently mutated genes, that is Notch1 and MYH6. So Notch1 had Notch1 mutations was more found in TFK samples, and in COA samples, MYH6 mutations were enriched, and also Notch1 was more expressed in the endothelial cell and MYH6 in the cardiomyocytes. So this inference we got from the current, where we have only a single cell data. And even our genome variant size was very small. So now we are planning to extend this to a large cohort at the genomic level. So we have samples from 10,000 CHD cases. So we'll be using that and developing once again the candidate gene list, and possibly by then, and we are also carrying out the CHD single cell analysis in our lab. So possibly by then that will be ready and we'll have better insights, and this study will form as a pilot study for us. So this is the overall paper. It is published and it is open access. So you can access this paper at APS site. It is in Physiological Genomics. And before ending, I'll quickly like to thank my PIs, Dr. Bakrum and Dr. Mohammeduddin, Al Jalila Foundation for funding my postdoctoral fellowship, Mohammed bin Rashid University, which is my parent university, and thanks to PCGC and CDDRC for giving me the PCGC and CDDRC fellowship. I thank all my lab mates, both academic and CATG, and all my fellows at the Al Jalila floor. Thank you.
Thank you, Richa, for this great talk. Let's just wait for some Q&A. All right. So maybe I can start asking a simple question. So do you think what we have here is a collection of rare disorders that is hard to converge into one cell type? Is it the case? Because it is true variability in the phenotype?
CHD itself, the data which we have is from many different types of CHD. It's not, so that's the reason of the heterogeneity, I believe. It's not one specific type of CHD. So that's what I would like to do in the further analysis. Even the single cell data which we have is from TOF and HLHS. It is not homogeneous. So right. This heterogeneity is kind of expected.
So they kind of give you Notch and MYH6 have 2 different pathways, right? But
yes,
and you see kind of heterogeneity there, but it's also visible at the phenotypic level. But at the end, it's not converging for endothelial and cardiomyocytes for all the CHD, though. There is a subset of it, is you are seeing.
Yes, when we combine all the genes, then we are seeing mostly in cardiomyocytes, because that is a majority of cell type which is present. But there's, as such, no, it is not converging for all the genes.
So one of the question is from Timothy. So I'll read out the question. If CHD is a heterogeneous disease, do you find clusters of variants that tend to associate with given phenotypes? Could this be used to define different subtypes or diagnosis of CHD?
if I get your CHD is a heterogeneous disease, that's correct. And
is there any
if I get your question,
cluster of variants that kind of tend to cluster
connected
together
to one
produce the
particular
same phenotype? Yeah.
Yes, so a cluster of variants connected to one particular phenotype, for example, say TOF. Right? Am I getting the question right?
Yes, yes.
So that's possibly the inference from this work also, because Notch1 was specifically from this particular data showed that Notch1, most of the TOF cases had Notch1 mutations. So possibly Notch1 mutations are related to TOF, and then they are specifically present in endothelial cells. So we are planning to extend this work and to see in the larger scale how different CHD genes behave, and if we can define the phenotype using the variants.
Okay. Another question from Lily is, can you also detect these mutations in single cell RNA-seq data? Do you see the mutation?
If you're talking about the Hill et al. Data, they have provided that information. They didn't have Notch1 MYX mutations. And if you're talking about a different set, yes, that can be done in the analysis stage. So
I think she was mentioning if the data has the single cell data that produced from this study also have the mutation.
No, the single cell data don't have these mutations. No.
Okay. Thank you, Richa. That was a brilliant talk, and I want to thank all the participants for this great inspiring talks for the experimental session. We have learned from smell to neuron to heart disease. So our next session will be moderated by Naomi Habib, and so please do not forget to join our next session by clicking join now. Thank you so much.
Thank you. I think we are continuing with the same Zoom, right?
Yes, everyone remain
okay,
here.
So
Thank you so much.
So just a small clarification, remain here. We're continuing straight away with our next session, which will be involving highlights from single-cell research in the Middle East. And our first speaker for today would be Itay Tirosh from the Weizmann Institute, and he will share with us his insight on spatial organization of glioblastoma. So Itay, the floor is yours.
Thank you, Naomi. Thanks for the introduction and for the invitation to speak. Do you see my slides?
Yes.
Okay, great.
Yeah, presentation mode. Yeah, perfect.
Great, thank you. So over the last few years, we've been studying very extensively the diversity of cells within glioblastoma tumors. These are the most common primary brain tumors. And initially, we used single-cell RNA sequencing, and what we found was that in every particular GBM tumor, we see different states of the cancer cells. The cancer cells tend to exist in these 4 different cellular states in which they resemble different types of normal cells. So one set of cells resembles oligoprogenitor cells, so we call these OPC-like cells. Another group of cells resembles neural progenitors. This is what we call NPC-like cells. We have cells that resembles astrocytes. And finally, we have a group of cells that resembles mesenchymal cells. And these mesenchymal-like cells can be induced both by interaction with the macrophages, and we've showed that macrophages induce the cells to transition to a mesenchymal state, and also by hypoxia, which is something that I will get back to. So after we defined the main pattern of diversity within those tumors, more recently, we've been very interested in understanding how are these states organized within the tumor. Can we understand, based on their location in the tumor, what dictates this diversity? How do those cells interact with one another and with the other cells in the tumor? For example, the immune cells and the normal glial cells. And so to address all of these questions, we've been using spatial methods, and mostly we've been focusing on 2 spatial technologies. We use spatial transcriptomics for more specifically 10x Visium. And the main advantage of this is that we get pretty good, pretty high quality data that is unbiased, that includes the entire transcriptome and allows us to discover new patterns of gene expression. But the limitation is that this is not really single cell resolution. When we count the number of cells per spot, we see a median of 8. So to complement this, we also use spatial proteomics using the Codex machine, or what is now also called PhenoCycler. And here we do really get single cell resolution, and we also see the protein levels, which is an advantage. But the limitation of the Codex is that it relies on a limited set of antibodies. And so we worked quite hard to establish this panel of antibodies that allows us to capture and to annotate all of the cellular states and all of the cell types that we are aware of in glioblastoma. And I will switch back and forth between these two methods to try to together explore the pattern of spatial heterogeneity in glioma. So I'll start with the Visium analysis, and we have quite a lot of data, 32 different samples. And from this, we were hoping that we could derive directly from the Visium data all of the cell states and all of the cell types that are present in those tumors. So, of course, each spot contains multiple cells, but despite that, we speculated that if we are able to identify all the patterns of variation within those tumors, because each state varies in its abundance between spots, we would be able to identify all of the cell states. And so the way we analyze the data is one sample at a time. We use both Leiden clustering and non-negative matrix factorization to find all of the major programs of gene expression that vary in those tumors. We then want to focus on those patterns that vary across multiple tumors, not just in one specific tumor. So we cluster all of these programs, and we focus on each cluster that contains multiple programs. We collapse all of these individual programs into the consensus, or what we call a meta-program. And each such meta-program corresponds to either a cell state or a cell type that appears to be present in those tumors. And indeed, we were able to identify all of the components that we are aware of. So in blue, you can see the meta-programs at the top that corresponds to non-cancerous cell types like neurons, oligodendrocytes, macrophages, vascular cells. In orange, you can see the states that we already previously found by single-cell RNA sequencing, so we can rediscover them in the spatial data. And also at the bottom, you see these additional 3 states for which the signal in the single-cell data was very weak, so we did not define them as new states. But in the spatial data, we see them more clearly. And this is presumably because cells in those states are perhaps a bit rare, but they are clustered together spatially. So in the spatial data, we see more signal for them. And we're still trying to better understand the meaning of these 3 different states. So after we defined all of these cell states and cell types, this is how the data looks like. We assign each spot to the dominant cell states. And these are just examples from 2 of the 32 samples that we have. And using this data, I now want to focus on describing 3 different modes of organization that we see in glioblastoma. The first one, and perhaps the most obvious one, is that for every particular state, we see that cells in that state tend to be clustered together. So the tumor is actually composed of small patches, such that in each patch there's mostly one state. And we can see this both among multiple spots in the Visium data, but also within a single spot. And in order to look at what happens within a single spot, we have to switch to the Codex data. The Codex does no real spots, but we can ask what happens for the cells that would have been in the same spot in the Visium data. And what we find is that in most of the spots, in 64% of the spots, there's just one cellular state that dominates that spot, meaning that it covers more than half of the cells in that spot. So even though there are 14 different states, for every particular spot, there tends to be just one dominant state. And if we ask how many spots are dominated by 2 different states, meaning 2 states are sufficient to cover the majority of the cells, then this goes up to 99%. So almost all spots can be explained by just 2 main cellular states. So this is one evidence for the clustering of cells in a given state. We can also ask what happens to adjacent spots. And so now we're switching back to the Visium data. And to address this, we defined a measure of spatial coherence, which is basically asking for every individual spot, what fraction of the neighboring spots immediately neighboring that spot are also in the same state. So for this red or purple state at the top, you can see that only one neighbor is in the same state, so this is low spatial coherence. And for the bottom, for the yellow state, this is the hypoxia state that I already mentioned, we see very high spatial coherence. So almost all of the neighbors are of the same state. And as you can see in this example, we do see differences between different states. Some states are more coherent than others. But when we do this analysis across all of the samples, the main difference that we find is not between states, but rather between different samples. So in some samples, overall, the tissue seems to be somewhat organized, and different spots seem to be in the same state as their neighbors. While in other samples, we see much less organization. And so we refer to these as structured and disorganized samples. And we were trying to understand what is the difference between them, what dictates whether a sample would be structured or disorganized. And the main difference that we find is, again, this hypoxia, this mesenchymal hypoxia state of cells, which you see in this dark yellow or orange color. And so samples that have a lot of hypoxic spots in them, not only are the hypoxic spots clustered together, but also the entire sample seems to be more organized. And this suggests that hypoxia is really functioning as an organizer of the entire tissue. Here you see additional examples, multiple structured samples. Again, you can see in all of them this dark yellow color, this hypoxia, and a number of disorganized samples. And rarely we find samples in which part of the sample seems to be structured and the other part seems to be more disorganized. This is also an example from the codex data. Again, the sample on the left seems to be quite organized with this blue area at the top and purple area at the bottom, but the right sample is quite disorganized. So this was the first level of organization. Each state tends to be clustered, and this is much more prominent in some samples than in others. I want to switch now to the second level of organization, which is to ask what is the association between different states. More specifically, we ask for every pair of states, how much do they tend to be appearing together in the same areas of the tumor? And we can ask this question using different methods at different levels of resolution. So, for example, within a given spot of Visium, we can ask how often do we find both of the states together, this yellow and light blue in this case. We can ask for one spot and its adjacent spots. How often are spots in one state adjacent to spots in the other state? And we can ask for a particular region in the tumor, and we can explore different sizes of regions. How commonly do we see those two states in the same region? And so we have multiple types of analysis to address this. We also use both the Visium and the codex data, and with all of these independent analysis, we defined state-to-state associations. And there's a lot of different associations that are specific to one method, but what we are mostly focused on are those associations that are seen quite consistently, that seem to be very robust. For example, we see that astrocyte-like cells, AC-like cells, tend to reside proximal to OPC-like cells very commonly in most of the analysis that we do. So this is a very robust association. And we also find negative association, certain pairs that almost never appear together, much less than expected by chance. But let's focus more on these positive associations. So now that we see these individual associations that are quite robust between different states, we ask whether all of these associations together give rise to some global organization. Can we learn something more general about how glioblastomas are organized? So to address this, we generated this graph in which each node is a state and each edge is this robust association that we find across multiple methods between states. And as you can see, this kind of automatically generated graph naturally separated all the states into 5 different layers. And when I say layers, what I mean is that all of the associations, all of the edges are either within a layer or between adjacent layers, but they never skip a layer. So what is the meaning of these 5 layers? The first one at the left is the hypoxic niche. This is where we see all of these mesenchymal hypoxia states. The second layer are the states that tend to be adjacent to hypoxia, the ones that seem to be more prominent close to hypoxic areas. The third layer includes multiple things, but we speculate that these are the states that are most important to resolve the hypoxia, for example, the vascular cells and the macrophages, an altered metabolism state, and so on. The fourth layer includes cancer states that are more sensitive to hypoxia. So these are presumably the states that require more oxygen. These tend to be neurodevelopmental cell states. And finally, we see the last layer, which is composed primarily of the normal brain cells, the oligodendrocytes, the astrocytes, and the neurons. And this is an example
we're
of these.
reaching the end of your time.
Okay, thank you. This is just an example from codex data in which we see in one particular section all of these different layers. So to summarize, we use the Visium data to directly derive all of the meta-programs, all of the cell states and cell types. We see that each particular states tend to be clustered. This is seen both within spots and across adjacent spots, but the degree to which this happens varies between samples, such that some samples are structured and others are more disorganized. And this is linked to hypoxia. Structured samples tend to have much more hypoxia. We find a consistent set of state-state associations, and when we consider all of them together, we find this global organization with 5 layers of states. So finally, I want to thank everyone that were involved in this work. This work was led by Elisa, Noam, and Ruven from my lab, and you see them on the right here. And this was a collaboration with Mario Suva from MGH and with Maria Neidert from St. Gallen. And thank you for listening, and I'll be happy to take questions.
Thank you, Itay. That was really fascinating. We have several questions, so we'll try to go through them quickly. So start with one online. So Yotam Avidar Constantini is asking, when a single sample from a patient is disorganized, do you expect the whole tumor to have the same phenotype?
So to some degree, yes, we expect those tumors to be more disorganized, but it is also reflecting sampling within the tumor, because if we take a sample at the very core of the tumor, they tend to be more structured. And if we take a sample from the same tumor that is really the periphery, it tends to be more disorganized. So this is both a property of the tumor and a property of where we sample exactly. It's a combination.
So this goes with a question from Timothy Little, who asked if the tumor disorganization is associated with patient outcomes.
So yes, it is, but I'm still not sure to what degree the organization is what determines the outcome, or rather this might be a byproduct. For example, if you know the glioma field, then you would know that IDH mutant glioma is kind of the most distinct form of glioma from the others, from the IDH wild types. And IDH mutant glioma tend to be much more disorganized. So we see this association with survival, but I'm not sure that the organization itself is the important feature that determines that.
Mm-hmm. So for sake of time, there are more questions online, but we'll go with a question by Yusuf, and you'll ask just answer the other questions online.
Thank you, Naomi. Thank you, Itay. This is really fascinating. So you did mention variation between samples, but I didn't catch if these samples are like primary or recurrent glaucoma samples. Could that be contributing to this variation in this state-specific clustering that you see, and could they be linked to some sort of a network or synaptic organization as well?
Yeah, so all of these samples are primary. and we have yet to analyze recurrent samples. They might be a little bit different, that's true, but we still see a lot of diversity between the primary samples.
And just if I might, so could that state-specific clustering be manipulated through some sort of interventions?
So if we can change the hypoxia, we would, the degree of hypoxia, we would speculate that this will change the organization, but we have not been able to show that yet.
Oh, thank you.
Thank you, Itay. And we'll move to the next speaker. So our next speaker is Nasna Nassir from Mohammed bin Rashid University. Tell us about the single-nuclear RNA sequencing, reviews the MAPT expression dynamics and correlates with tau pathology in human brains. So go ahead.
Thank you, Dr. Naomi. Good evening, everyone. Today I'll be discussing our latest findings on Is my screen visible?
Yeah, now we can see it. Thank you.
Yeah, I'll be discussing our latest findings on how single nuclei RNA sequencing is revealing dynamic changes in MAPT expression and how it is correlated with tau pathology in progressive supranuclear palsy. So a brief introduction on what progressive supranuclear palsy or PSP is. This neurodegenerative disease syndrome is clinically recognized by progressive instability, Parkinsonism, and cognitive decline. And diagnosing PSP is identified by characteristic features like neurofibrillary tangles or NFT and tau-positive astrocytes. Interestingly, there are several mutations in MAPT, which is known to cause a form of familial frontotemporal degeneration alongside with tauopathy, and this has been identified in both sporadic and familial PSP cases. So focusing on MAPT or microtubule associated protein tau, there have been earlier reports saying it's aggregated in neurons in diseases like PSP. So our hypothesis questions on how the cytopathology alters MAPT expression in different brain cell types and how it could pave a path to have tau as a potential therapeutic target. So our primary objective was to quantify MAPT expression across different cell types in both control and PSP affected brains. And we leveraged techniques like single nuclei RNA sequencing and RNA-scope to deeply understand the relationship between tau pathology and MAPT expression. So delving into the methodology that we use for the study, for single nuclei RNA sequencing, nuclei was isolated from human frontal cortex in 3 control and 3 PSP cases. And the cell barcoding was done using 10x genomic single cell 3' kit, and were sequenced on NovaSeq 6000. So for a disease like PSP, identifying MAPT expression in individual nuclei was critical due to its cell heterogeneity. And alongside single nuclei RNA sequencing, we did RNA-scope, which helped us in quantifying and visualizing the expression of MAPT in situ. So to start our analysis, we did dimensionality reduction and cell clustering, for which we employed, started with 10 principal component and clustered using k-means algorithm and visualized using UMAP. And next, we annotated the cell type using known brain cell marker genes from curated from literature and cell marker database. By looking at the expression of these marker genes in differentially expressed genes of each clusters, we categorized it into like different neuronal and glial subtypes. So this was critical for further downstream analysis. So our initial observation was an increase in number of excitatory neurons and decrease in oligodendrocyte in PSP cases, which could be an indicator of PSP pathology, which is yet to be explored. And tuning into the single nuclear RNA result, we observed that MAPT expression was associated with only specific cell subtypes exclusively present in inhibitory neurons, oligodendrocyte and astrocyte. So this prompted us to focus more on these cell types to further associate role of MAPT in these cell types to connect to PSP or any other neurodegenerative diseases. So next, we did RNA scope analysis and found a positive correlation between MAPT expression and tau pathology. Specifically, MAPT expression in neurofibrillary tangles in frontal cortex and basal ganglia were explored. And this showed a strong link between MAPT expression and tau pathology. So in conclusion, we proposed a pathway of how tau maturation happens and how the pathogenic tau is being released into extracellular space for neurons, oligodendrocyte and astrocyte. And we propose a therapeutic intervention where it can be targeted to targeted removal of misfolded tau, and decreasing MAPT expression could help in slowing down or halting the progression of PSP. So I would like to take a moment to acknowledge our collaborators at UHN, Dr. Garber and team, and my colleagues at Mohammed bin Rashid University, Dr. Mohammad Uddin, Awab and Shehzad, for their expertise and in contributing to the research. Thank you for your attention. I'm happy to take any questions.
Thank you so much. Maybe I would start with a quick question, although just a second. Oh, sorry, I had a problem with my microphone. I was wondering if you were able to see a link between the neuronal and the non-neuronal cells in terms of signaling or within the data as well.
We haven't explored the signaling pathways.
Mm-hmm.
For this work, we were mostly focusing on the MAPT expression.
Mm-hmm.
So others are on the way. We are exploring other. What are the?
Because it seems to be so highly linked, right? Especially, at least based on what you've shown, it seems like neuron and astrocytes at least have this very strong coordination, or maybe I'm wrong.
They are. So we are prominently looking at astrocyte and other markers, apart from MAPT. We started with MAPT as there are a lot of literature supporting link of MAPT and PSP. So right now we are looking at non-hypothesis driven, looking at discovering new markers localized with the neurofibrillary tangles in astrocyte. That's in progress. Yeah.
Great, thank you. So I'll ask another question from the Q&A. Sarah is asking if your samples were paraffin embedded or fresh frozen.
These are fresh frozen samples.
Great, thank you. Yeah, and so we will transition to the next speaker. So we have a talk by Bruno Guillotin from the New York University in Abu Dhabi. Bruno, go ahead.
Thank you very much, Naomi, for the kind introduction. So, all good. Okay, so thank you very much to the organizer for giving me the opportunity to present my work. especially because I'm a plant scientist. So today my job is to excite you away from neurons and cancer cells into the world of roots and especially in crops. So I'm working in NYU Abu Dhabi in the lab of Kenneth Birnbaum. And in the lab, we're really interested in multiple types of crop species, such as corn, sorghum, and millet. And you're pretty much aware that due to global warming, all these different crops are actually facing harsher and harsher environments. And so they have to adapt to these stresses. And what's quite interesting is that even though these crops are extremely important for food consumption in the world, they don't all answer to the stress similarly. Some are actually more resistant than others to the same stress. And what's interesting here is corn is a very heat and drought susceptible plant. And so farmers need to add a lot of water to the field to get a decent harvest. What's interesting is while sorghum and millet are quite closely related from corn, they're much more heat and drought resistant. So in our project, we're trying to understand how is that possible and what
Bruno?
Yes? Yes?
Can you speak into your microphone a little bit better? It's a little distorted.
No worries. Is it better now?
Yes,
think
much.
That's
Yeah.
much better. Thank you so much.
No worries. Thanks. Okay, so what we're trying to do here is to understand how different crops can respond differently to different stresses, to understand what are the genetic pathways that are involved in this resistance. And so to tackle this problem, we work on root development, because roots have been the long forgotten part of the plant that have been neglected by plant breeders and during natural selection and agriculture. So here on the right, you see a wheat variety here, an agricultural one, and you see in red the extent of the root in this variety. And you see it's very, very small and shallow on top of the soil. But when you look at the same species, but a wild variety, you see this massive root system extending deep into the ground. And because during domestication, people were only looking at the upper part of the plants and not really looking underneath. And so because roots are so important in providing water and nutrients to crops, we wanted to investigate how these roots are adapted to stress, or how we can use different variations to understand how they adapt. And so roots are extremely complex organs. They contain multiple different cell types, have gradient of maturations, and so on. And so we wanted to understand which cell type may respond to stress. Are there certain cell type more prone than others? But we were facing quite a strong challenge, because in crops, there were no single cell RNA-seq map of roots. And more importantly, in multiple crop species, we have no idea of the cell-specific markers expressed in this cell type. So we started our project a few years ago, and were able to publish the first single-cell RNA-seq roots map of corn. And during extensive validation and mutants analysis, we're able to identify every cell type in there. And so we wanted to see, can we use our knowledge we have in corn to transpose it to other crop species? And so we work on sorghum bicolor because, as I told you, it's a much more heat and drought tolerant plant than corn. But importantly, it has diverged fairly recently. And so they share a lot of genes together. And as an outgroup, we use Teteria viridis, which is a cousin of millet, and which is becoming also a much more important model system in different labs. However, we had no idea of any gene expression in these roots before. So we wanted to see, can we use our knowledge from corn to transpose that into other species? So to do so, we generated single cell and single nuclei RNA-seq of these different species using 10x genomics technology. And we perform a common integration using the R package Seurat and using autologous genes that have been mapped between species. And so in this big UMAP, each dot is actually a cell or a nuclei from either maize, sorghum, and Ceteria. We then predicted the different cell identity using markers we have in corn. And we wanted to validate this prediction because I was only relying on corn markers. And so to validate that in sorghum and Ceteria, we went for a systematic validation using in situ hybridization that allows us to see marker gene expression in the root. So we developed a whole mount in situ hybridization technique using in collaboration with Doris Wagner in UPenn University. And so here's an example of an endodermal markers. So a gene that Sarat was giving us to very specifically express here in sorghum. And you can see here a translational root section of sorghum. And you see in magenta, the probe related targeting these genes very nicely highlighting a single cell layer in the center of the root. And so that is actually the endodermis. And so we systematically validated that in our system. Here's an other example is sorghum with a xylem marker. So xylem are these pole of fibers that conduct the sap in roots. And here's an example in corn in cortical markers, and you see how specific the expression is. So to go a step further, we also validated our data set using spatial transcriptomic, or what we can call molecular cartography. So here in gray, you have a slice of root from corn longitudinally cut. And we were able, working with Resolve Bioscience, a company in Germany, to map up to 100 genes on these slides and to further validate our single cell RNA-seq data on a single slide. So at the end of this study, we were able to fully identify up to 15 different cell types across 3 species, and most importantly, identify hundreds of new marker genes in sorghum and Ceteria that are of primary interest for plant breeders. And so with all this data, now we can dive into it, we can have fun and see what's going on. And so one of the first questions we had in response to stress is, are there certain cell types that the transcriptome is very conserved across species, like cell type doing the same jobs, while maybe other cell types are maybe more prone to adaptation and emerge new mechanisms across species? So to test this hypothesis, we developed using a meta-neighbor approach, a transcriptome divergence score. So here on the left, on the y-axis, you see all the different cell types we identify in our single cell. And on the x-axis, you see the transcriptome divergence score of corn or sorghum in respect to Ceteria. And so what you can see is that on top of this curve, of this graph, cells in G2M, so cells in active division, have a very low divergence score, which makes sense, like cells divide using the same transcriptional pathway across species, even across kingdom. It's a very conserved mechanism. So it made sense that cells were actually in low divergence. And what you can notice is that epidermis and trichoblasts, which is a type of epidermis, also have a very low divergence score. So it's very likely that these cell type epidermis are pretty much conserved across species. However, when we go down the graph, you can see that stela or endodermis, which are more inner part of the root, have a much more divergent score and could evolve additional mechanisms. And what was very interesting for us is that the most divergent cell type, columella, which is the very tip of the root, was the cell type with the most divergence. And so we identified, I don't have time to present today about it, we identify transcription factors that were actually switched between sorghum and Ceteria to maize in the columella, and we think are linked with domestication and improving nutrient accumulation. So from this study, we had set the ground truth about the different marker genes in the cell types, and we showed that we could use one reference data set to infer cell identity on other species. So now we have all the tools to actually stress our plants, which was our primary question. And so that's the ongoing project we have in the lab. So we're growing corn, sorghum, and millet in different stress conditions, especially heat and drought. And what we want to see is which cell type in the stress responds most to stress. And is there species-specific adaptation to stress? We want to identify a gene expression pathway in sorghum, for example, that's very heat or drought tolerant, that we could maybe transpose into corn or give to the breeders some insight. So to finish my talk, I wanted to talk about another project which is ongoing in the lab, involving endogenous species of UAE, the mangrove tree. And so here on the left, you have a beautiful picture of Jumeirah Island in Abu Dhabi. And on the right, you have the mangrove tree, which compose like 100% of this green you can see there. And so our question was, how do mangrove adapted to salt? And from the literature, what we know is that there are two main adaptation strategies. The first one is mangrove filtrate the salt out from the root. So salt will never enter the root. You have a very specific barrier that's built involving very specific cell type. And here you see this ring of very shiny green, which is actually suberin deposition in one single layer of the root. So here's the translational section and transversal section. And so this suberin is actually very hydrophobic. And so all the water and ions need to go through cells and not between cells to go into the root. But we have no idea of the genetic pathway and the genes involved in that. The other strategy is salt get in, but is exuded from the leaves. So you see on the leaf like crystal of salt that are exuded by specific cell type. And that's another project we invested in. And so very quickly, what we're doing is we grow mangrove seedlings in no salt condition and in salt conditions. We take the root to do single cell RNA-seq and perform a common integration. We identify the different cell types, and what we are currently analyzing is which cell type responds to salt, and can we identify transcription factors and gene conserve across crop species for integration in a further screening selection for breeders. So my time is up. I wanted to thank my PI, Kenneth Birnbaum, for his amazing support, our postdoc in NYU Abu Dhabi, Alia Hessin, and our technician who's been working with us for many, many years now at TIG, and our team in New York. Thank you very much.
Thank you so much. This was great. Thank you for opening up the world of plants to us. I'll start with a question from the chat. So I'll just read. Timothy Littles is asking, thank you for the talk. Great to see plants represented. How do you select the parts of the roots to sample from the original specimen? And is there a reason to believe that the roots may be different between, for example, shallow and deep roots?
So that's a very developmental question that we are also investigating. So to simplify our system, we're only taking the first roots that's coming out from the seed and taking like just the very young parts to have the same developmental stages across species and to be able to compare them more thoroughly. We have an additional project where we take within the same plant different roots, so the primary, secondary, laterals at different stage of development, and trying to understand how roots are actually of different root type actually different transcriptionally. And so we can find that certain root type like laterals would have specialized cells in the sap transport, for example, which are very specific transcriptomes compared to primary roots. So if you want to compare across species, you also need to take into account the diversity of root type and just take the same roots across.
Mm-hmm. Interesting. So another question, an interesting technical question from Lala Agavardi. Thanks for the talk. How do you define comparable divergence scores among different cell types, given the different size of gene modules for each cell type? Also, non-orthologous gene change of TF regulatory role along organism, evolution, etc.
Yeah. very simple question, right?
Yeah, it's a good question. It's a hard one.
So no, we are tied by the autologs for sure. And so for now, we're only looking at autologs across species, and the non-autologs one, or the one we cannot map, are more difficult to assess across species, of course. What we are doing is this meta-neighbor approach, which takes about 5,000 most differentially expressed genes between species, and just compare in one by one, give a score of expression, of correlation, and just compare transcriptome across cell type and across species. So if your data is good enough, your integration for one species, one cell type in one species, its transcriptome should look like the same cell type in another species, and not another cell type in the same species. I don't know if I'm clear, but it's a complex analysis. So meta-neighbor approach has been used in multiple systems, and it's a quite robust one once you have the right genes. So if you're interested in this integration, I suggest that you look into this method. It's a bit, it's quite technical and statistics, so I'm not sure I can explain fully here.
Yeah,
Sorry.
It could be out of our scope, but I think the general explanation was very clear. So last question, I hope I'm pronouncing the name right. Shahzada Ibrahimova is asking if you looked whether genetic difference of mangrove trees reflect on irrigation.
Oh, no. So that's a very specific question. So we are starting the project, and what we are trying to do is to understand between species that have adapted different strategy to salt, how cells actually have evolved. And maybe there is one cell type involved in this salt resistance in one species, and it will be another cell type in another species. And so regarding irrigation, I'm not sure if I understand. I would say that what's interesting for us is that mangrove tree specialize different cells to actually get air into the root, because it's growing underwater, so it needs to breathe. So there are specific cells that carry the air from the surface towards the roots. So that's another project we are maybe investigating in the future to understand how airflow and cell type are important for air, which is very important for crops, because when crops are flooded, if the roots are adapted to breathe better, then they can survive this extended period of flood. So rice, for example, has adapted similar strategy to grow underwater with the root system.
Okay, thank you.
Thank you.
And we'll move to our next speaker. So back to the mammalian world. We'll talk Yasmin Demerdash from the German Cancer Research Center at Heidelberg Institute. Will tell us about hematopoietic stem cell and progenitor cells. Yeah, we can see our slides. Just unmute yourself.
Okay, so now you can hear me well? Hello?
Yes, perfect.
Perfect, yeah, thank you. 1 second. Yeah, so I'm very happy to be here today to present part of the PhD work that I've done in the lab of Dr. Marika Essers from the German Cancer Research Center here in Germany. So yeah, let's dive right in, and I will start with a brief introduction to the topic. Okay. So yeah, in our normal day-to-day life, as we're sitting right now, we're exposed to several agents with which the body is regularly challenged. But thanks to our immune system, it takes care of that and protects us by fighting against the invading pathogen. But this, of course, leads to a high consumption of the blood and immune cells. And humans lose approximately 1 trillion cells a day. So yeah, the recovery and maintenance of a balanced hematopoietic system depends on the self-renewing and division of hematopoietic stem cells, which are residing in the bone marrow. and they just like proliferate in order to maintain a steady state response. HSCs are multipotent, meaning that they can give rise to all lineages of blood cells throughout the lifetime of an organism. However, they are quiescent. They are mostly residing in a G0 state of the cell cycle. Nevertheless, upon inflammatory stress, such as following severe bleeding or following bacterial and viral infection, the quiescent HSCs start proliferating as they directly respond to many of the pro-inflammatory cytokines released upon those stress conditions. And this response serves to control emergency hematopoiesis and presumably to restore the system by replenishing the mature cells which have been lost. Yeah, and thus enhancing the host defenses. So as I've showed you now that inflammation and stress do have a significant impact on our hematopoietic system. And this is where actually my PhD work came into play, as it specifically focuses on studying inflammation and specifically acute inflammation on the hematopoietic stem and progenitor compartment. So in my project, we've used mice as our model organism, and we've used interferon alpha as our inflammatory stimuli, as our lab actually was one of the pioneers in investigating the effect of interferon alpha on HSCs. Already now more than a decade ago, Marika have showed that when you inject mice with interferon alpha, take out the bone marrow after 16 hours, you start seeing the HSCs proliferating as they go into the G1, G2 SM, as indicated here in those FACS plots. But this only provides a snapshot of the response. And what we now more appreciate is that this is more of a dynamic response where there is a window of time between when the cells sense the treatment, respond to the treatment, and we know that the cells go back to their quiescent state after the response is over. And in the lab, we conducted experiments to obtain specific time points that can effectively capture those phases. So we already now know that at 3 hours, the cells start sensing the treatment by upregulating their an ISG called SCA1, among other ISGs, of course. However, at 3 hours, we don't see any proliferation yet. At 24 hours, we see that the ISG level go back to normal, whereas the cells start proliferating at its maximal response. And at 72 hours, we see the cells already going back to their quiescent state. So most of the studies in the literature have focused on the response of HSCs. However, the impact on more downstream progenitors is still not clear. That's why here in this project, we were mainly interested to understand how adaptations would happen throughout the whole HSPC compartment. So this is the classical hematopoietic hierarchy. And this is the HSCs residing at the top of the hierarchy. And underneath, we have the multipotent and oligopotent progenitor. And this is the compartment where we're actually interested to study how interferon alpha impacts. So what we did was going for an unbiased approach where we wanted to investigate the molecular changes in the whole compartment. So we did a single cell analysis where we've injected mice with interferon alpha in a control and then sacrificed the mice after the time points where we know that the HSCs sense, respond maximally, and then go back to the quiescent state. We then isolated the bone marrow and then we perform cell hashing in order to still retain the information from a single biological replicate as we've used 4 mice per time point. And then we perform FACS sorting. But in FACS sorting, we gated for a broad gate, like lineage negative c-KIT positive, in order to capture a wide spectrum of the HSPCs. And then we perform the samples for sequencing. And this actually project we've submitted the paper for revision, and it was a collaboration with a group from Dr. Lali Hagverdi from the MDC Berlin, where together Lali and Bridget, they were driving the computational analysis for this project. So initially what we did was, after performing the sequencing, we've obtained 4 different data sets and identified several clusters. And most importantly for today's talk, I will only be highlighting the HSCs and the myeloid clusters because those, just like I will mostly focus on those. So what we first wanted to investigate the heterogeneity of the interferon alpha response. So what we did first was scoring each cell for an expression of a set of interferon stimulated genes where we know that those genes are upregulated after interferon alpha treatment. And what we can see here is that the largest change in ISG expression is seen at the 3-hour time point, and they go back to recovery in all the compartments at 72 hours. And most notably, we see that the HSCs have the strongest ISG change following the treatment compared to more downstream progenitors. what we did next, so here we can see that all clusters do sense the treatment, with HSCs showing the strongest response. So yeah, next, we wanted to evaluate the transcriptomic changes upon interferon alpha treatment. So what we did was we performed comprehensive differential gene expression analysis, where we systematically examined changes in the expression within all time points and over all cell types, and summarized these in the following heat map, where we categorized the response genes into 14 groups. Those were either globally responding genes versus cell type specific responding genes. Yeah, and among the genes which were globally changing, those genes were enriched in processes related to translation, metabolic, viral, stress and interferon processes. And as an example, those two groups had genes which were mostly changing in the myeloid compartment, and those were mostly enriched in myeloid function and differentiation processes. Whereas those group of genes were changing mostly in the HSC clusters, and they were mostly related to an immune response. And also notably from this heat map, we can see that HSCs have the highest number of response genes, indicating that they have the strongest transcriptional change following the treatment. So yeah, so basically with this analysis, we managed to identify global and cell type specific analysis. But here what we're missing is the direction of change or the dynamics of the gene expression. So in order to do this, we wanted to do a pseudotime analysis. However, using the conventional pseudotime analysis, this didn't really work for us, because in a normal pseudotime estimation, they would assume that the proximity of cells is a measure of time. But in our case, we have a recovery system where we know that the cells at 72 hours, they actually tend to go back to the recovery state. So in that case, a proximity of two cells cannot be used. So we needed a new computational approach, and that's where Lali and Bridget have developed this response pseudotime, where we now can order the cells and convert discrete time to continuous time. And from this analysis, now we managed to also do, sorry. I'm sorry, I think yeah. Okay, so we categorized the response genes into 16 patterns based on their expression dynamics and pseudotime using hierarchical clustering. And each of those patterns represent a cluster of genes with similar expression dynamics following interferon alpha treatment. As an example, pattern 6, 7, 8, and 9, those are genes which have a rapid response, like they respond rapidly and then they go back to their quiescent state. And those were mostly enriched in interferon processes. Whereas we had other pattern of genes which failed to recover back to their quiescent state, and those were genes mostly related to mitochondrial translational processes. And we had other group of genes which actually had a sustained downregulation, and those were related to myeloid differentiation genes. So now we also see that the interferon responding genes show a variety of expression patterns with different biological functions. So now we're having actually two analyses, one which have cluster-specific inflammation signature, and another which have the dynamics of the gene pattern. So what we did next was combining both analyses to have this heat map, where we can now know the response genes specific to each group, how do they behave in time, and where do they fall in their pattern. So as an example now here, for example, these two groups were specific to the HSCs, and we now know that most of the HSC specific response genes are actually following this rapid dynamics in time. And another example is for myeloid genes. We see that the response genes for the myeloid clusters were mostly following this downregulation or sustained downregulation in time. And this was very nice to see that different clusters have different response genes, and they have also behaved differently in time. So the response to the time reveals HSPC, gene dynamic heterogeneity during the inflammation induction. So having the signature of sustained downregulation of myeloid function genes made us interested to see also if this would impact on the cluster size or the abundance of those cells. So we performed, so basically wanted just to check if this reduced myeloid transcriptional program would impact on the progenitor compartment size. So we did an abundance analysis, and what we actually see that we have one subcluster of the myeloid progenitors which have shown a sustained downregulation or sustained decrease in their size. And this was actually very interesting for us because this is not a classical thing you would see, because usually upon inflammation you would see a classical emergency hematopoiesis, and in our case we have seen downregulation of myeloid function genes. We've also seen abundance of the decreased abundance of the myeloid cluster. So this was very interesting to see actually with interferon alpha. And we next started to investigate the causes of this myeloid depletion. So maybe the cells are moving out to the bone marrow, or maybe those cells are dying, or it could be a mix, and also the cells, there's an impairment of the stem cell myeloid differentiation. So in order to investigate the movement of the cells out to the bone marrow, we've checked cells in the periphery, such as in the spleen. We first checked the cells in the bone marrow, and we've seen actually that, as we see in the single cell analysis, we also see a decrease in those progenitors in the bone marrow. And checking them in the spleen, we actually see the similar picture, that the cells are also being depleted from the spleen. So in the blood, there was no also signs of increased size of the myeloid progenitors. So yeah, maybe moving out to the bone marrow isn't the reason from our current analysis. And we've checked for cell death. So we checked, came back to the single cell data, and we checked for pro-survival genes, and we've seen that those pro-survival genes actually tend to go down. So we did an experiment where we've used mice that lack apoptosis machinery, and we've injected mice like the back knockout mice and wild type mice with a treatment, and sacrificed the mice after the 3 time points that I've indicated earlier. And what we see is that both the wild type and back knockout mice, they also have a reduced in the myeloid progenitors, which could maybe, apoptosis couldn't be the reason. However, this doesn't exclude other pathways involved in cell death, such as necroptosis and pyroptosis. So we came back to the single cell data, and we've checked for necroptosis and pyroptosis gene signatures. And interestingly, in the myeloid progenitors, we've seen an increase in the pyroptosis gene signature. So maybe pyroptosis could be playing a role in cell death of those progenitors. As a third investigation was checking if there's an impairment of the stem cell myeloid differentiation. And by checking a set of myeloid transcription factors, we already see it at the HSC level that there's a downregulation of the myeloid genes. And when moving to the myeloid compartment, just to check for monocytic neutrophilic differentiation, we've seen that myeloid progenitor 1 and 2.
I'm just interrupting you, so you should start wrapping up.
Yeah, okay. So we see them at the myeloid progenitor 3, which was actually have the sustained decrease in abundance. They show a decrease in the neutrophil differentiation. And in order to see how this is reflected in the blood, so we actually checked neutrophils and monocytes in the blood, and we can see that there's a sustained downregulation of mature neutrophils in the blood. So this now brings me to the summary. So here, by combining single-cell RNA-seq with interferon stimulation and adding the dimension of time, this helped us to examine the heterogeneity of the response. As we can see that we have globally responding genes and also cluster-specific responding genes. And using our response pseudotime now, we can also see how the different genes behave in time. And we can also associate those dynamics into cluster-specific responses. We also see from the abundance analysis the decrease in the myeloid progenitors and signs of increased cell death by increased pyroptosis signature. There's an decreased myeloid differentiation, which was at the end reflected as a decreased myeloid cell production in the blood. And at the end, I would like to thank you so much for listening to my talk. And of course, I would like to thank my group, the stress group, and my supervisor, Dr. Marika Essers, and Dr. Lalia Verdi for their supervision for this project. And Bridget for we're a great collaboration, and I'm happy to answer your questions. Thank you.
Thank you, Yasmin. Really great talk. So, okay. We'll start with the first question on the online. Another job is asking how can you relate this to a disease model? How can you relate results?
So thanks for your question. So there are many viruses which are known to increase actually interferon alpha. And there was already one study which showed that when you give a vaccine with MTB, that this has an opposite effect to the trained immunity. So they found that the myeloid progenitors on the long term were having reduced response to the infection, and also patients which like systemic lupus erythematosus patients, they are known to have a neutrophil dysfunction, and they are also known to have increased interferon alpha. So maybe studies later can try to correlate or relate how the interferon could impact actually at the end on the neutrophil for those patients, and if by blocking maybe the interferon, this will enhance their neutrophil responses.
Interesting. Thank you. Maybe I'll add one more question of my own. So I really love your dynamic clustering and how you really kind of tailored out all the different responses, but I didn't completely understand how you ordered the cells along a pseudo time. Was it completely without the information of time based on their cell programs, or if you can provide more information?
Let me get back. So to be honest, for the pseudo time, I didn't really dig deep into how the Lali and Bridget did it, but I think Lali is also in the audience, so maybe if she can answer this question better than me.
Lali, she's not. Let's see if she's available. It's okay. I can ask. Yeah, go ahead.
Yes, we actually use the time information. So it's a linear regression model that tries to capture real time of each cell. I'm sure Yasmin would be also, but maybe because I am here, you're a bit.
I just thought that you best answer this question. Yeah.
Thank you.
Yeah. So do you have any predictions for what would be the upstream regulators of these programs that can reverse the effect? So like transcription factors?
So we didn't really check for that. Maybe further analysis would be nice to have, and yeah, but also it would be nice to check for later time points to see actually how this effect, until when it's sustained, or maybe it's reversed back after 72 hours.
Okay. Yeah, thank you.
Thanks.
So that would actually conclude our session. I would like to thank all the everyone attending the session today. We still have our closing remarks. I do want to add a kind of my own that I hope that such meetings would really advance science in the Middle East and help us foster more collaborations. And I especially want to thank anyone attending who are affected by the latest events in Israel. I can attend from my own experience that after suffering such great loss of loved ones and actually waiting for kidnapped to return, and still so much anxiety over the state of war we are in, it's really not trivial to attend a meeting. And personally, I'm really heartbroken and lost for words from seeing so many children suffering these days. I see my own children afraid for their lives, like so many other Israeli and Palestinian children. And I'm really hoping for and praying for better times. And hopefully through science, we can bring something more positive to this region. So thank you. And our session is done. And Kristen, who's the next?
Yes, so I think John and Yusuf will come on for closing remarks for day one. Thank you so much, everyone.
Thank you. Thank you so much, everyone. Thank you, Naomi. And I reiterate our thanks to everyone, all the speakers and moderators for really excellent sessions today. It was really interesting. We had a broad overview of the HCA, the researchers, and also the range of talks ranging from humans, mice, tissue, different tissues and cells, both in health and disease. Bruno's talk on single-cell genomics in plants was a nice distraction from a mammalian system. We thank him for that. And also special thanks to the team actually taking care of the organization, particularly the team in the US, in the East Coast, Christine and Bridget for staying awake and engaged all night. So the recordings of today's sessions are live on the event website. So just go to the agenda page, and each relevant session there is like a watch record on button for anyone who missed any talk. Feel also free to share the link to the event website for those who didn't have the chance to attend. And we look very much forward to tomorrow's session. We look forward to seeing all of you. And John, do you have any comments you might want to add?
Yeah, I have very little to add other than to say on behalf of the HCA Executive Office and the HCA leadership, thank you to all of our speakers and moderators. Thank you to everyone for attending and for your interest. And we look forward to seeing you tomorrow for day 2, beginning at noon Gulf Standard Time.
Thank you all.
Thank you, everyone.